Okay.
So, welcome everyone. Thank you for taking out time and joining us here today. We are from LifeGen Bio, and our speaker for today is Dr. Avinash Mishra. I would like to give a brief introduction about him and today's topic as well, and the topics that we are going to cover. So, to start with Dr. Avinash Mishra's introduction, he has completed his PhD from IIT Delhi. He has gained more than 5 years of research experience after PhD. He has various skills under his hat, including computational structural uh, biology, drug design, Linux, statistical analysis, molecular dynamics, neural network, project management, business development, and you have to just go on. Like they do not stop. Later, he has also authored 12 peer review publications, one book chapter, one workshop article. He filed one Indian patent titled Polypeptide Sequences and Composition thereof. He is an editor at Scientific Reports, Nature Publishing Group. He was awarded Indo-Australian Gold Fellowship for postdoctoral position in the year 2017. That is a great achievement. Congratulations, sir.
Thank you.
He did a collaborative research in the area of drug discovery, machine learning, and computational protein structure as well. He headed a team to conduct a big BIRAC project awarded by Department of Biotechnology in 18 months. That is also an amazing achievement. So we are glad to have you here. So with the warm welcome, we welcome you here. And later to talk about what we are going to cover in this webinar is that you must have read it on the flyer as well, but I would like to brief you, those who are new here. The first topic would be ligand-based drug design overview. Second would be chemical database for small molecules. The third would be 1D, 2D, 3D descriptor calculator using online installed tools. Then would be QSAR modeling. Then database creation of for QSAR. Later would be create your own QSR model using R. QSR model testing and validation using R. Later would be overview on pharmacore design. Then the last would be hands-on pharmacore screening and modeling tools. So here we are, and we are going to have a very fun and interactive session with Avinash Mishra here. So we welcome you, and I hand over the platform to you, sir.
Thank you, Unmeet. Thanks a lot.
Yeah, welcome.
So, guys, just before starting the session, I hope now you can see my screen now. Yeah?
Yeah, Dr. Avinash, we can.
Yes, we.
Yeah.
Okay. Okay. As Unmeet said, that lots of things we are going to cover, but let me tell you that what we are planning is this webinar is more on that there is a hands-on training and workshop we are going to organize very soon after this webinar, and there we will practice more on ligand based drug designing. So some of the topics that were just discussed and mentioned, that will be much more covered in the workshop, because we cannot cover the hands-on and everything in just 1 hour webinar session. But what I will try to give, I will try to give you a idea that what is ligand based drug designing, what are different techniques that people are doing for ligand based drug designing, and that what are the tools that could be used, how they can be used for ligand based drug designing. So these are the things that I'm going to cover. So this is much more like a like a teaser, or maybe you will get some brief idea, and then if you want to go for the next hands-on training, there you will get the real crux of ligand based drug design. That's the plan. So let me just start the session, that what is ligand based drug designing and why it is important, and what are the tools and what are the techniques that people are doing. And this I would just try to cover in next 40-45 minutes, and then I will leave you for asking some questions if you have. The coordinator of this webinar will, coordinate the question answer session, and then I will answer some of your questions. So before uh, before going for very specifically about the ligand based drug design, let's just talk about drug. you know when we say drug, so what exactly we refer to. So one of the definitions says that a drug is any substance other than food that when it goes inside the body, it makes physiological changes. So that is one of the definition, and it's quite true for many substance. So anything which is not part of your general food and you are taking it for making some physiological change can be called as drug. But most of the times when we say drug in field of research, then actually we are referring to pharmaceutical drug, and these are the compounds that can be used to treat, cure, prevent, diagnose a disease, or to promote well-being. So that is what we call a drug, what a researcher like us calling a drug, and that is the pharmaceutical drug. So this is one of the example. Paracetamol, we are very much familiar with this drug, and we can get it over the counter. And here you can see a chemical skeleton of of paracetamol. That's it looks very simple, but we all know that how important it is, and how crucial this particular drug is. So what exactly there in the chemical molecules, and then how we can design some new molecules, some better molecule for curing or preventing some of the diseases, you know. And that is what I'm going to give you a brief idea in this webinar. If you talk about the sources of drug, so from where we are getting the drug, so of course, the prime source or one of the ancient source is the nature from where we are getting the drug. And we all know that there are many natural compounds that has pharmaceutical capability, that has therapeutic capability, and then we are taking these either directly or extracting the therapeutic chemical compound from the natural source, and then we are taking it to the lab, and then the next source that we say is the lab, where we exactly go and synthesize the molecule. So these are, the two verticals of getting the drugs compound, getting these therapeutics compound. For a structural biologist like me, what is drug? So when we say drug, what exactly we are referring to? So we are referring to a compound. So I'm just reading the first bullet. A compound which in general call as ligand that binds to a biological target. In general, biological targets are protein, but in some of the cases it could be, like RNA or DNA also. But most of the times it is protein. And the function of this compound, the ligand, is to either activate or inactivate the protein function. When it is activating the protein function, we call it agonist. When it is inactivating, it is we call it as antagonist. Now, if you want to understand this protein and ligand interaction, because they have to interact with each other for making the physiological changes that we discussed. And actually, this interaction is necessary for triggering all kind of physiological changes. So this is the first step. The interaction is the first step. And to understand it, you can take it the example of lock and key, though it is a very crude example, I would say, because it's a very rigid interaction, lock and key. But just to understand, you can consider the biological target protein as a lock, and the drug as a key, which is responsible for opening the lock. So there is a physical interaction between lock and key, and then there is a function of a key to open the lock. So you can, take it as an example that proteins are lock and drugs are keys for those locks. And this binding, the protein and ligand binding, is mostly stabilized by many kind of interaction forces. like electrostatic, hydrophobic, hydrogen bonds, and there are many more. I mean, definitely in the next session workshop, we will take all these forces one by one. We will calculate it, and we will know that how to calculate and which force is much more important and why it is more important. But here you can just understand that these are the key interaction responsible for drug in a protein. Now, there is I just wrote it old school of drug discovery, but believe me, it is not that old. It is, people are still following it. And where they are doing high throughput screening, where they are checking lots of molecule in one go. And these are experimental. All the steps that I have written in this slide are experimental steps. And first they do the high throughput screening, where they are checking the activity of 100 and 1000 of molecule in one go. And there is a robotic infrastructure for that, and then people are doing it in higher capacity, and then they get some hit compound, and then they optimize it, hit to lead, and then they further optimize the hit lead compound, which is called lead optimization. And then they get something which may call as a drug candidate, and then these drug candidates goes for clinical trial. And once they pass the clinical trial, then we can call it as a drug molecule. So initially, they are just hit, then they are lead, then they are drug candidate, and finally they are drug. So this is like, the whole pipeline. But because I have a direct experience of working with pharmaceutical company, Astra, and where I found that there is a high chance of failure, where the chemistry department, they are generating many hundred and thousand of molecules, and checking it through the pipeline, and at the end they found that no, there is no chemical compound giving us the desired activity. So there is a high chance of failure, and we all are aware with it, that this drug discovery is, it's quite a serious discipline. Now, what we can do as a computational biologist or a computational chemist, that we can add something called in silico, which is when you are try to simulate some of the stuffs of experiments on your computer, so that you can reduce the chance of error, or you can reduce the chance of risk. So that is, initially it was in vitro and in vivo, means on cell, or on enzyme based assay, and then on animal in vivo. And then later on, people have also introduced in silico, just to improve the accuracy or just to reduce the risk. And now, and that's why we are, now talking about in silico drug designing or computational drug designing. If you classify in silico drug designing into two broad sections, you will find that there is something called structure-based drug designing, and very soon we will arrange a workshop for the structure-based drug designing also. And there is something called ligand-based drug designing. So for structure-based drug designing, here the structure refers to a structure of a protein. When you know the structure of a protein, when you have the structure of a protein, and then you design a drug molecule for that particular protein, then it is called structure-based drug design. But in many cases, we are not very sure about the target. We are not very sure about the which protein actually it is targeting, or maybe sometimes we are sure that yes, this is targeting this particular protein, but unfortunately we do not have the structure. So from where we are getting the structure? We are getting the structure by solving the XRD, which is called X-ray crystallography. By this technique, or NMR, by these techniques we get the structure. But NMR and X-ray crystallography is not very easy. It's a very sophisticated method, and in many cases we don't get the structure of a desired protein. So that's why we also have something called ligand based drug designing, where we don't need the structure of a protein to which we are targeting, but we what we need is a set of molecules that already showed some activity. for that particular protein, and we will use those set of molecules, known molecules, for designing something better than them. So that is called ligand based drug design. So here in this session, or uh, in this webinar, and also the follow-up hands-on and workshop later on, we are going to cover this ligand-based drug design. And in this ligand-based drug designing, one of the technique which is majorly used, and some of you might be aware with this technique, is called QSAR. And this is the major topic that we are going to cover, QSAR, which is called quantitative structure activity relationship. So I will discuss it more. And then we also have some technique called pharmacophore mapping. So that is also we are going to cover in brief. And there is something also called fragment based, which is not a part of this workshop or this webinar. Now, let me just give a simple definition of ligand-based drug designing. When we say ligand-based drug designing, what exactly we mean? So, I'm not covering structure-based drug designing. Maybe, definitely, not maybe, definitely in future we will cover it. So, in this session, we are going to cover ligand-based drug designing. So ligand-based drug designing is an indirect approach to facilitate the development of pharmacologically active compounds. Definitely, we need something which is active by studying the molecules that interact with the biological target. So, as I said, here we don't need the structure of the biological target, but what we need is a set of compounds which have already shown some activity uh, in the experiment for particular target, and we will use those compounds and design something better. How we can do it? Let's go further and try to find out that what we can do in this ligand based drug designing, and especially in QSAR or pharmacophore. So a structure of known ligands are collected. So when I'm saying known ligand or drugs, it means that somebody, like you and me, uh, we have done some experiment and we found that this particular molecule is showing some activity. Now let me tell you first that what is activity. Many times we heard this word, that activity of a compound. So activity can be defined in many ways, but one of the very primitive way to define activity of a compound is that what is its binding with the particular protein, and what is its IC50, something which is called IC50. So IC50 is nothing but it's a inhibitory concentration. 50% inhibitory concentration means that the concentration of a compound that is required to inhibit the function of a protein by 50%. Okay, so when the IC50 is low, it means that the compound is good. Definitely, because we need drug in a small amount. I mean, nobody wants to take any pharmaceutical drug in higher amount, right? We take pills, just a small pills, and that is also with formulation. So, which means that the activity of that particular pills is sufficient in a very small concentration, and that is what we refer to activity when we are saying activity. So let's first collect some known ligand or drugs from different chemical databases. There are chemical databases where they are reported, and we can take it from there. We can collect the structure, we can collect the activity, means IC50, or sometimes KI. KI is also another parameters, which is the very similar to IC50. So that is also depends that the what is the concentration to inhibit that particular protein. Fine. So now once you collect the chemical compound, means chemical compound collection means the structure of the compounds, or uh, the formula of the compound, or the smiles of the compound. And once you collect it, then what we believe that there is a structural similarity or pharmacophore matching or substructural search method. So what it says, that there is a rule that similar chemical structures behave similarly, right? So if there is a similarity in the skeleton of a compound, among the compounds, then their activity would also be similar. That is, the hypothesis, or that is like the backbone of ligand based drug designing. Structure activity relationship we have to establish. And what I mean by structure activity relationship is that how the structure of particular compound is related with its activity, so that in unknown case, when you get a new compound and when you have the structure but not the activity, you can predict its activity. Okay, so this is what we are doing. We are collecting the compounds, we are establishing the relationship between its structure and its activity, and then we start making the models, and then we start using the models for predicting the activity of a unknown compounds. So so what we need, so these are, I'm not putting lots of time on these slides. Maybe there are much more interesting slide later on. So what we need for ligand based drug designing? So definitely we need data, right? So when we are saying data, it means the chemical information, structures of a molecules. It also refers to their corresponding IC50 or corresponding activity. Okay, so these are the data. Then we process the data, and then we use the data. Then the information that we extract from the data can be used for the further prediction. So let me tell you that how we can do all these things. Okay, don't worry if you are not very clear with some of the points, but once I move to for the slides, it will be much more clear to you. So now, as I was saying that, the similar molecule exhibits similar properties. So here you can see that there are three molecules, and by the structure you can easily detect that these are very similar, right? They are almost like 90-95% similar, but there are some differences in some functional group. You can easily figure it out, right? So what we believe that because they are 90-95% similar, so there is high chance that they will behave similarly. So behaving similarly in drug context means that their activity would be similar, or their IC50 or their binding with particular protein would be similar. That is like the, as I said, the backbone of a ligand based drug designing. Okay, so now this is just a snapshot saying that you collect data, you apply different kind of models, and now people started using machine learning also for this purpose. We started with linear regression, and linear regression works fine in current scenario also, but because we have also have sophisticated machine learning method, so we can also apply those machine learning method for making the model. How? Let's see. I will tell you that how we can do it. So here, just see the pictures on left hand side. So as I was saying, the IC50. So one IC50 is experimental IC50, which is on the x-axis. So from where we are getting this IC50, we are getting it from the chemical databases or from the known information. And then there is a predicted IC50. From where we are getting these predicted IC50 is using the models that we create. And then we see that how good my model is. And there are certain metrics or evaluation method by which we can find out that how good my model is. So one of the simplest technique is that you just put IC50 experimental and IC50 predicted on the chart and see how good they are linearly correlated, which is reflected by the R square. So R square is a regression coefficient for this model. So here in this case, R square is 0.9, which means that it is highly correlated, which says that if you are predicting using this particular model, whatever the model is this, then there is a high chance that you will lie somewhere close to its experimental value. So for unknown set, you can use this model and predict the activity. Okay. And if you are predicting, you are very sure that my prediction value of IC50 is very good. I mean, very close to the experimental. And then ultimately you have to do the experiments. But what you can do is, in place of doing experiment for, let's say, 1,000 molecule, you can only do the experiment for let's say 100 molecules, and only for those molecules for which you are getting a good predicted IC50. Yes. So that is why we are doing this LBDD. So it is not 100% replacement of experimental technique. Okay. So that's we have to understand that this is not 100% replacement, but this is something which can contribute in the drug designing, which can lower the risk, which can save time. So that's why we are using it. So as you have seen in one of my slide, high throughput screening. So in high throughput screening, we usually use, let's say, 1,000 compounds. But before going for the experiment, if we do similar kind of QSAR and predict the IC50 for these 1,000 compounds, then maybe we get to know that out of 1,000 compounds, only 10 molecules are working good. I mean, their IC50 is really good. So let's go for only 10 compounds in the experiment. That will save a lot of time, money, effort, everything, right? So, but the question is how we can build this model. So that is the main um, concern, that how we can make the model. So as I said, for making the model, you need to collect the data. And what is the data? Data is the chemical compounds and its corresponding IC50 for known. Okay. We will only make the model on the known data set. So it's like on the known data set, we build the model, and on a known data set, we will predict the model, predict the IC50, predict the activity using the model. So just I'm just reading it for you, that QSAR is a statistical method to find relation between chemical or physical property with the activity of the molecule. So I think by now you all are very clear that what we are doing in QSAR, we are establishing a relation between the property and the activity, or the property and the IC50. When we say property, it could be chemical property, physical property, like molecular weight, log P. I will discuss more on that. There are many packages available. Generally, I'm using R because R is, first thing, R is freely available. And the second thing is that R has lots of liberty for you to actually modify, change your own model. Okay. And that is what we are going to do in hands-on training later on. And the end point of the bioactivity, which is the IC50. This is what we want to predict, or we call it as a predictor. IC50 is a predictor. And what are the features? The features are the chemical properties. Now, just steps in QSAR: identification of active ligands, right, from the known data set, then identification of the suitable descriptors. So guys, don't get confused. People are using the word like descriptors, physical properties, features, all are same. So identification of suitable descriptors, that is the next step that you have to choose, and then establish a mathematical relation between these descriptors and the activity, and to construct a model. So that is what we are doing in QSR. So this is just a pictorial representation of same. So activity is a relation, a function of properties. Okay. And we take the known experimental IC50 and then set a relationship of known experimental IC50 with all the properties. And then that is what giving us the model. And then using the model, we predict the activity of unknown compounds. Right. Now, one thing is definitely QSAR, but there are related terms also, like QSPR, where P means properties. Let's say I'm interested in calculating the solubility of molecule. but I don't want to do the experiments. Okay, so you can also design and develop a mathematical model for predicting the solubility. So that will fall under QSPR. Okay, so this is just a different jargon, but the base of that will be same. Again, you will collect the compounds, known compounds, and then extract the features or descriptors, establish a relationship, make a mathematical model, and then calculate a predictor. So in this case, my predictor is not IC50 but the solubility. Similarly, you can also develop something for toxicity for a compound. Let's say I want to just find out that what would be the toxicity of my compound. Similarly, you can do the same. Now, sometimes people say that, okay, I understand SAR, which is structure, activity, relationship, but why it is quantitative, means why the Q, for what the Q stands for. So definitely Q stands for quantitative. And why we are saying quantitative? It's very simple, because we are using the mathematical modeling stuff. We are taking it as a numbers. Okay, so that's why it is quantitative. So when we say the activity is bad, we just cannot say just the activity is bad or good. We give some numbers that, okay, the activity is, let's say if we are dealing with IC50, we say the IC50 of the molecule would be 3 nanomolar or 30 nanomolar. So definitely 3 nanomolar would be better than 30 nanomolar. Okay. And why we need QSAR? As I said, that the synthesis, because before checking the compound, you have to synthesize the compound. And believe me, synthesis is not very easy process. It's time consuming and it's expensive. So if we can do something prior to synthesis, prior to these experimental testing on my computer, why not to do this and then just go for those compounds which are actually meaningful, which are actually going to perform in the experiment. So that's why we need QSAR. Now, when we say features, descriptors, properties, no, and the structure, activity, relationship, what exactly these features are? Okay, so what exactly these features are? No, and people have categorized these features into many category, and one category is just in front of you. There are some hydrophobic features. Okay, you can. Uh, I hope that you are getting my points. This step is after collecting the data of known ligands. Okay, now let's say that you collected data for 30 ligands for which I have the experimental activity or experimental IC50 or experimental Ki value. Now what I will do, I will calculate the features or the properties for these 30 compounds. And what kind of properties I would calculate? It could be categorized as hydrophobic properties, or electronic properties, steric properties, and quantum chemical properties. Now the question is, why these kind of properties? So just by looking at these properties like partition coefficient, Hansch substitution constant, or hydrophobic fragments, so these properties are actually responsible for their activity. That's why we need to calculate. Okay, now suppose, just take an example. Okay, I'm just because many of you are from BTech, so I just want to make it a bit simpler. Let's say that there is a guy, or let's say that there are hundred of people standing in front of you, and question is that you have to detect, or you have to find out just by looking at them, that who are athletes among all these 100 peoples. So this is the question. Okay, that who are the athletes. Okay, so in this case, athletic is like my activity. Okay, being athletic is my activity. Now I have to predict, or I have to find out that who are the people among these 100 people are actually athletes. Okay, so how you can estimate by looking at them. So you just go to their or you just observe their features, like how tall they are, or how fit they are, or maybe if it is for very specific sports, let's say for basketball, then the height matters. So these are like some of the features that are actually responsible for um making them athlete, right? So similarly for chemical compounds, these are the features which are actually responsible for their activity. And maybe when we will do the hands-on, we will go to these properties, understand that why these properties are important, what this property is actually saying, like the partition coefficient. What partition coefficient is saying, that how much of your compound is soluble in octanol and in water, that is the ratio of that is your log p. Similarly, what is substitution coefficient or constant? Substitution constant is that, let's say if you are replacing one functional group, let's say you are replacing hydroxyl functional group with carboxyl function group, then what will be the effect? Okay, so what will be the effect on its activity? So that is your substitution constant, and people have already done experiments, and they have already calculated these constant, and we can use it for our QSAR model. Okay, and similarly like the steric, so steric is like what is the bulkiness of the compound. Hydrophobic, that the non-polar characteristic. Electronic is something which is related to the electron cloud of the compounds. Steric, as I said, that the bulkiness of the compound. And then there are like quantum chemical properties, which are most of the times related to your charge or your energies. So these are something which is directly impacting the activity of a compound. That's why we need to calculate before going for building model. Now, as I said, activity is a function of properties. In this slide, here you can see that the activity is a function of properties. Now I'm just replacing the properties with the exact properties, the term properties with the exact properties. So one of the initial model by Hange in 1969, this is that time we were not using the computational method in such a rigorous manner to build a model like these days we are using. But at that time, what he found, that the activity of a compound is related to these three different features. One of the features, don't go by the first name, like the Hammett substitution constant. You just understand substitution constant, which I said, substituting one functional group with another functional group. Then there is Taft's steric constant. Again, don't go the first name, but the steric effect. And the third one is also another substitution constant. So these three, he has done some experiment on a set of compounds, and he found that there is a direct relationship between the activity of a compound and these three parameters. Okay, so then it is very much possible that if we get this equation where C is the concentration, or log 1 by C, you just consider it as an activity. Okay, so it's very much possible that using this sigma, pi, and es, you can calculate the activity. Once you establish this relationship on a known data set, then you forget about the known data set, and then whenever you encounter with any new molecule, that time you need not to do the experiment. What you can do is you can just use this mathematical equation and then find the activity. Okay, that is what the QSAR. This is what the QSAR. So this is like the simplest one. and now we have grown up and we have we can build our own QSAR model. Now, the most important thing in QSAR is the what kind of properties you are selecting. Like here, Hans has selected only 3 properties, and he solved his own problem, whatever the problem he had. So it's not necessary that if you have thousands of parameters, your mathematical equation would be accurate. You have to be very choosy with the descriptors or the features. So maybe sometimes you need many features, sometimes you just need just a small set of features, so anything possible. So selecting these molecular descriptors are very important. Okay, so let's see one of the molecule which is nowadays in lots of news because of COVID, remdesivir. Okay, so when we say molecular descriptors, and this is the best word to use rather than physical or chemical properties or features, I think the best word is the descriptors. So descriptors are the mathematical numbers which describe your molecule. Okay, so this is what we say descriptors. So as I said, that initially we have the molecule. This is the molecule, right? Now from the molecule, we need to generate these descriptors or these properties. And these properties would be numbered, of course, because we are going to use mathematical equation, and mathematical equation will only understand the numbers. So that's why we need to calculate these descriptors in form of numbers. And then we represent this particular, let's say, remdesivir can be represented by a set of descriptors. Example, molecular mass, number of nitrogen atom, total charge, number of aromatic ring, radius of gyration, and so on and so forth. Okay, so this list can go for very long, and then all these numbers collectively can represent molecule remdesivir. Okay, then you need not to worry about the remdesivir itself, but you just see the numbers. And of course, when we are dealing with mathematical equation, we just deal with the numbers. So we get the molecule, and then from the molecule we get these numbers. These numbers are of course the chemical numbers. These are not like arbit numbers. All these numbers based on some specific chemical characteristic of a molecule, but we calculate it, and then we forget the molecule, keep these numbers. So this is one of the book. I think if you guys are interested, you can go ahead reading this book. And it's saying, the jargon, the whole definition is a little bit complex, but I'm sure that you can understand because I have already described it. That the molecule descriptor is the final result of a logic and a mathematical procedure which transform chemical information encoded within a symbolic representation of a molecule into a useful numbers, right? That's what I said. Okay, so you take the molecule and then you calculate these descriptors, and then you use these descriptors for whatever the mathematical modeling you want to use. Now, generally, these molecular descriptors are categorized, into 3 classes: 1D, 2D, and 3D. Okay, so one-dimensional descriptors, two-dimensional descriptors, and three-dimensional descriptors. Okay, so one-dimensional descriptors are those descriptors that can be calculated directly from their formula. I hope that you guys know what is smiles. Um, Please read. If you don't know, just it's very same way the way we write the chemical formula. This is also a kind of a string which represent your molecule. Okay, so those features that can be directly calculated from the formula or the smiles would be considered as a one-dimensional molecular descriptors. Now, some descriptors need the 2D structure. Okay, so like here, right? Or if you remember that similar molecule behaves similarly, there are three molecules. So those are the 2D representation of a molecule. So 2D representation of a molecule, and that can be used to calculate some of the 2D descriptors. And these 2D descriptors are normally called topological descriptors. Okay, so it describes the topology of a molecule. No, that what is uh, or what is the skeleton of a molecule? How the molecules, atoms of a molecule are there in two-dimensional space. So that is one of the property, one of the 2D descriptors, a very well-known descriptor is called Wiener index. And I will just describe that how to calculate these Wiener index. And believe me, it's not that complex. Once you know the trick, you can calculate it using your own hand also. I mean, you don't need even computer or calculator. So, and then there are something called 3D descriptors, for which we need the three-dimensional structure of a molecule. So a molecule definitely has one-dimensional representation, two-dimensional representation, and three-dimensional representation. But in reality, every molecule are three-dimensional. That we all know. So if we use three-dimensional structure for calculating the descriptors, so those descriptors are called 3D descriptors like polar surface. Let's go one by one. I think 1D is very easy. Uh, you know, oh, okay. So this is what I was referring to. So earlier I said say a similar molecule behaves similarly. Now the same thing I'm saying in slightly different way, that the molecules with similar descriptors behave similarly or exhibit similar activity. Okay. So source of the descriptors, from where we can get these descriptors, one is experimental, of course. Means if we have experimental informations like log P solubility, NMR shift, you can take it from the database, you can take it from literature, you can just extract it. You need to just do a curation, and the other way is calculate theoretically using some program. Okay. So these are the two ways for calculating the descriptors. So 1D, like the molecular mass or number of carbon atoms, number of oxygen atoms, these are very simple. I mean, you all can find it out. Now, I'm just jumping to 3D, sorry, 2D, 2D descriptors. So I'm not sure that how many of you have seen a structure file or the chemical molecule file. But this is the format of a chemical molecule file, SDF, in 2D format. Okay. Now you can see that this is the X coordinate. This is the Y coordinate, and we don't have anything on the Z coordinate in this particular file, and that's why it is called 2D SDF file. Guys, if you are not aware with these files, we will do this. Okay. So that's why I said earlier that this webinar is much more on giving you um, that what we are going to actually do in the next session. Okay. So okay, Janish, there are lots of people who are waiting, almost like 50 people are waiting. Or is it, are they waiting or?
No, no, Dr. Avinash, everyone is in the room itself. So we have just stopped the waiting room thing.
Okay, okay, okay,
Yeah,
fine.
everyone is here directly.
So, okay, so this is the 2D SDF file. In the hands-on session that we will conduct, the plan is for next weekend. There we will know how, from where we can get it, how to download it, and how to use it. But for time being, let's assume that this is the 2D SDF text file. This is, for computer, it is like a text file, and it is representing the XY coordinate, and that can be transformed into this structure, right? Means, because in 2D SDF file there is a connectivity information also. Using that information, we can draw this graph. a simple molecular graph, which atom is connected with which atom, and then we'll finally end up with something like this. Now from, so this is the same example. We have files, 2D SDF file. From SDF file we can make a simple molecular graph, and that simple molecular graph can be later on transformed into a matrix, right, which is called connectivity matrix. Okay, just be a little bit more focused, because I would explain. I cannot go for each and every descriptor, but I'm giving you the idea that what is the idea to how we actually come up with a 2D descriptors, and how we can design our own 2D descriptors. Okay, so now from the simple molecular graph, you can see that there are six atoms. Okay, 1 to 6, and that's why we can have 6 by 6 matrix. Okay, so in the matrix on the right hand side, you can see that there are 6 columns. Fine. Okay, and also 6 rows, right? So each columns is your each atom, right? So we have 6 atoms, so we have 6 number of column and 6 number of rows. How they are connected, that we will put in as a value of this matrix. So like, let's say 1 is connected with 2. In the graph you just see that 1 is connected with 2. So that's why in 1 to 2 the value is 1. 1 means connected. Here 1 means connected, and if it is connected with 2 bonds, we put the number 2. If it is not connected, we put the number 0. Okay, I think it's very simple, very easy. I mean, you can easily make this connectivity matrix. Now I will tell you why I'm making this connectivity matrix. Once you make, before that, there is also something called topological matrix, and it is also very similar to the connectivity matrix. So in connectivity matrix, if you see, this is the connectivity matrix on upper side. This is the connectivity matrix. We are just saying if it is connected and how many bonds are there in the connection. So if it is, there is 1 bond, we write 1. If there is 2 bond, we write 2. If there is no bond, we write 0. If there is 3 bond, we write 3. Okay, that is we call connectivity matrix. In topological matrix, what we say, same way, 6 by 6 matrix, 6 by 6 matrix, yes, 6 rows, 6 column. But now the values would be that if I want to go from one atom to another atom, how many bonds we cover, that will be the value. Let's say I want to go from atom number 1. So see the molecular graph, this graph. I want to go from this methyl group to this methyl group, 1 to 3. So we are crossing 1 bond and 2 bonds. So that's why 1 to 3, the value is 2. Now, if I want to go from 1 to 6, how many bonds we are covering? And in this, even the double bonds is also considered as 1. Triple bond also as 1. Just it's saying the path. It's not counting the number of bonds like the connectivity matrix. So from 1 to 6, 1, 2, 3, 4. We have 4 edges, right? In mathematics, we say edges. So that's why 1 to 6, the value is 4. Okay. So this is called topological matrix, and this is called connectivity matrix. Why we calculated it? For calculating one of the 2D descriptors, which is called Wiener index. Okay, and you can find out that there are many 2D topological descriptors based on the similar formula. You can you can innovate it, you can add something, and then you can modify the equation. But the Wiener index is saying that the sum of all the shortest distance, so here we are taking the topological matrix. So this is butane. Okay, I have taken the simplest one, butane, and made its topological matrix, and the formula is this. Okay, so dij is a distance between ith atom and jth atom. Okay, so if you apply this, and this is very simple. Okay, so if, let me just tell you one for one atom, and then you can of course calculate for other atoms also. Let's say I want to calculate for atom number 3. So my I, in this case, would be 3. Okay, and j would go from 1 to 3, and then every time whenever we are going from 1 to 3, we will take the number dij from this matrix. Okay, so for 3, no, so for 3 the values are 2, 1 and 0. 2, 1 and 0. Or the simplest way is you just add all the elements in one diagonal. Okay, all the elements in one diagonal. So 1 plus 2 plus 3 plus 1 plus 2 plus 1, and the overall is 10. Okay, so this is the Wiener index. Very easy. I mean, you can calculate for any molecule. Okay, and what it is saying, what it is representing, it is representing the topology. Now the butane topology is represented by a number 10, which is a Wiener index number. Okay, so that's what we are doing in mathematical descriptors. So this is just, I try to explain one descriptors. There are many more. Okay, and then, okay, you guys can calculate it for this molecule the same way I calculated for butane. You can try it later on. And now there are 3D descriptors. As I said, for 3D descriptor we need three-dimensional structures like the van der Waals surface, or van der Waals surface, or the accessible surface. Okay, so accessible surface is the surface which is exposed to the water, and for that we need the three-dimensional structure. For that we need the three-dimensional structures. The other three-dimensional descriptor is the, like the polar surface area. You know, this molecule, now this is the cloud, electron cloud, and here you can see that what are the atoms which are polar. So in this case we have oxygen, halogen, and nitrogen. These are the polar atom, and the area of these polar atoms are like one of the descriptors, which are called polar surface area. Okay, now there is something called autocorrelation descriptors, and I mean, believe me, this is also very easy to calculate. You know, sometimes these mathematical equations are a bit scary, but if you go to these equations one by one, you will find that it is not that scary. Now, like here, if you see, I'm taking the distance dij and adding them. All the diagonal element, I'm adding them and making it Wiener index. Now, in the autocorrelation, in addition to those distances that I'm adding, I'm also attributing some properties, you know, like let's say charge. Okay, so let's say charge. Now, so here, when I'm adding this number 1, I also go and see that what is the charge for atom 1 and multiplying the charge with this distance. So that is something which is called autocorrelation, and there are many formula for that. Okay, so here you can see this delta ij is same as distance, but wi and wj may be the charges. I mean, it could be charge, it could be mass, it could be electronegativity, and then you get a much more refined Wiener index. Okay, so many people have already formulated many equation, and then they calculated different type of um, properties. Now, tools for a descriptor calculation is like, you know, how you calculate. This is like the logic I just explained, or if you want to design your own descriptor, how you can design. But there are many tools available like ChemDes, Paddle, Moderate. These are the tools that can be used and what it needs your chemical molecule, and then it can calculate like 100 to 1000 different descriptors, 1D, 2D, 3D descriptors. So in hands-on session, we will use paddle. We can also use moderate, but it is Python based. So we will see that if you guys are comfortable, then we can take the moderate also. And then so the upper three tools are freely available. eDragon, now it is disconnected. Now people are means nowhere I'm getting this tool. And the other Codesa and Adriana, these are you have to purchase them. But I think Paddle and Moderate, I would say, if you can run these two programs, you would get sufficient number of molecular features or molecular descriptors. Now, once you get the descriptors, as I said, you need to develop a mathematical model between the descriptor value and the activity value. And for that, there are many tools. So as I said, I'm using R, but there are very specific tools for making QSAR model, like Build QSAR, Open 3D QSAR, and Biopsy. And these are the free tools. You can use it. And definitely there are some mathematical concept behind using it, but once you are familiar with those concepts, you can use it easily. Now, steps for model building, just I'm going quickly now. Is first is data collection, that is your molecule with activity, then feature or descriptor calculation, that is on the real, which is I think the real data set, because real data is not the molecule, but the real data is your descriptors of those molecules. And then feature cleaning, because some of the descriptors maybe are highly correlated. So these are the standard mathematical procedure that we follow. And then remove missing value, zero value, constant value. Then you divide the data into train set and test set, and then you create a model. One of the model is linear regression that we will see. And then of course there are machine learning models, so we will design some machine learning models also in hands-on. Then you evaluate your model, that how good your model is, and then you save your model, and then later on new data set you can use the model and predict the activity. That's how it goes. uh, in hands-on, just a few snapshot I'm just pasting here. This is one of the tool, paddle, if you remember. Here the second one, paddle. So paddle is molecular descriptor calculator. Here you just have to upload your molecule file and then the output file, and you can calculate 1D, 2D descriptors very easily. Okay. And then this is model, this is R Studio, because I'm going to use R Studio for creating the model. But one of the utility in R, which is called paddle, it has a graphical interface. So those people who are not very comfortable with writing codes, they can also use this one. This is also a part of R. This means you have to install R, you have to install R Studio, and then you have to install, this is one of the package called Rattle. And then Rattle, you can upload your data, and you can see that in this is a weather, some weather data, like location, minimum temperature, maximum temperature. But just replace, once you give your data set, it will be like, your all set of descriptors, like one from here to here, you will see your descriptor, and in one row you will see your activity. And then there is something called model here, and here you can easily, very easily, just a click, press option. It's very simple, even for experimentalists, that they can do it. Okay. Then how you evaluate your model. So there are certain matrices that people use, like least square fit or regression coefficient. People say that if it is greater than 0.6 or something which is called cross validation, we will, once we will make our own model, we will calculate these parameters on our own and see that whether my model is good or not. or based on these cutoff values. And also it says that you're not only on the set that you build your model, but also on the external data set. If your correlation is greater than regression coefficient is greater than 0 0.6, then only you can say your model is reliable. Okay. This is just a little bit about the pharmacophore. So pharmacophore is you know
So sorry to cut you in between, but I think the
yeah,
timing for the
time.
meeting won't allow us any further.
Okay.
So if you can wrap it up in 5 minutes, so
No, no.
that
I think so. The main focus for this webinar was QSAR. So I'm leaving it here because I think I explained a lot on QSAR. Pharmacophore, we will see it later on, maybe in another webinar sometime. And this is what the 2 days workshop that we are planning. Guys who are interested, I mean, there, you can do all these things that I discussed. QSAR data set extraction, and then descriptor calculation using Paddle, then model building using RStudio, and then model evaluation using RStudio. All these things we will do in the hands-on. Okay, so that's it. Yeah.